Okay, it worked. Before we start on the next chapter, I just want to sort of flag your attention a little bit to a couple of things that I've added onto the icon contents thing. Now, all of these things I'm talking about are supplemental. You don't have to go look at them. But if you're looking for something to take the course a little further or you want to take advantage of them, they're there for your use. I've put on 3 of the articles. We're okay. Well, we can do that. We can fix that. Okay. All right. I think we will go down on the lights. This is a little bit hard to see. Is that better? Yet? Is that too dark? Brighter? All right. More light? Okay. Anyhow, so we have under the supplemental reading heading several things that may be of interest for you. I've got 3 of the articles about the eggs cholesterol question. The JAMA article, Journal of the American Medical Association, is sort of the shorter form. It's their official editorial position. So even the American Medical Association has come to terms with eggs. That's sort of the short, quick version of it. And if you want to see what the actual work looked like, the other 2 articles are the primary articles that describe the egg farmers and the subsequent studies with a Western diet. Then the last link here is something you might want to be careful about clicking. It's 38 megabytes, so it's kind of a big file. But it's the wiki textbook that I mentioned, but as a PDF file. So if you want to load it onto your computer, you can download it from Icon and just carry it with you. I just mentioned that if you want it, but do be aware it's a 38 megabyte file. I thought doing it once and putting it on Icon would be kinder to Wikipedia than having 5 or 10 or more students making the PDF and then downloading it from Wikipedia. So, any other question about, someone else had asked about, there we go, practice exam 3. Someone had asked if we could post the practice exam today, and there it is. So we can thank Brenda for that. Are there any other questions about course matters or handouts or things like that? I have some queries out about the problems with downloading Google videos. I don't have a solution for you yet. I understand they are still playing all right. It's the download that's the question. And if I can resolve that, I will do so and let you know. Any other questions then about those? Okay. Then let's go to something substantive, some real stuff. Okay. So we finished on our last time together talking about lipids and lipid metabolism. Do people have any questions from the lipid chapter that they'd like to ask before we move on to nitrogen? Okay. So up until now, the kind of metabolism we've been talking about has really only dealt with 3 kinds of atoms most of the time, right? It's dealt with carbon, oxygen, and hydrogen. But those atoms alone don't provide enough complexity, enough sophistication to do the things that living systems do. We really depend on nitrogen as well. Nitrogen is key for proteins. Nitrogen is key for nucleic acids. So nitrogen metabolism will turn out to be sort of part of the mysterious inner core that makes living systems as complex as they are. So we're going to, as I just mentioned, that the nitrogen is essential for amino acids, essential for nucleotides. Now we're surrounded by nitrogen. 70% of the air we're breathing is nitrogen, but it's nitrogen gas, N2. And N2 is a very unreactive compound. It just is out there, and it might as well not be there for all the chemical interest it generates. It's why you fill automobile tires with nitrogen gas to make them last longer. To get it to be biologically useful, we have to fix it. That is to say, we have to take it from this diatomic gas and create a carbon-nitrogen bond somehow. And that process is going to be energetically expensive. We'll take a little bit of a look at how that happens. Once the carbon is fixed, we still need to get it into amino acids, we still need to get it into nucleotides, and rearrange it. We'll look at that a bit. Under some circumstances, there's actually excess nitrogen. Obviously, plants growing out in the wild don't have that problem all the time. But people living in Iowa who have a generous diet are likely to have more nitrogen than they need, and we'll see how that is handled. And then finally, when you have an excess of amino acids, those carbon backbones are then available for energy of various kinds. So that's the overview of where we're going to be going. So N2 gas actually has a triple bond, and that's a very inert bond, very difficult to make it do any kind of chemical reaction at all. Nitrogen availability is one of the major constraints on how fertile or productive things are. So when you're looking out in a field, nitrogen availability is key. That's why you have ammonia tanks out there to fertilize the fields with. One of the other constraints in the ocean is iron, but that's sort of a sideline. There are not very many organisms that can fix nitrogen gas into something organic. And those are going to be our friends, the blue-green algae. Someone has accused me of having a fixation with the blue-green algae or the cyanobacteria, but here they are again, fixing nitrogen for us, and some bacteria. And the methods that they use to do this are a really extraordinary piece of chemical engineering by living systems. There's an enzyme called nitrogenase. This is a diagram of one cofactor within nitrogenase. It's got some fairly odd chemistry to it. It has iron-sulfur centers, which shouldn't surprise us. We've seen iron-sulfur centers in redox reactions before. It has iron molybdenum. We haven't seen molybdenum in anything else. But you can see here how there's an iron molybdenum complex that is a very peculiar structure. And ferredoxin, our friend ferredoxin, which we last saw when we were talking about photosynthesis. This picture is just showing you some of the root nodules in clover, where some of the organisms that are capable of fixing nitrogen are protected from the environment through the nodules. To give you a sense of just how powerful and subtle nitrogenase is, when we do this process industrially, we need 400 degrees and 300 atmospheres. And blue-green algae and these bacteria can accomplish this at ambient temperature and pressure. Here's what the balance equation for it looks like. Nitrogen gas and protons and electrons. So you can tell that something's going to get reduced in here. And 16 ATP. So for 2 nitrogens, 16 ATP. And some water give us 2 ammonia molecules and some hydrogen gas and 16 ADP and inorganic phosphate. So the earliest step that we're going to be doing is not actually making the carbon-nitrogen bond. It's going to be making ammonia. But ammonia is a much more reactive compound, and it's much more readily incorporated into organic molecules. Ammonia as a gas does not last very long in the atmosphere, so it has to be made on an ongoing basis. This is the optimistic estimate for how much energy it takes to fix a molecule of nitrogen gas. There are some combinations in which it takes 20 to 30 ATP per molecule of N2 gas. So this is the most expensive biochemical process that we've talked about. And just to make life worse, oxygen can react at these centers in nitrogenase because oxygen is so reactive, and if it does so, then it poisons this complex and it is not doable again. And for this reason, nitrogen fixation is very restricted into where it can happen. There are certain habitats that can happen, very special chemistries that are involved, and so on. Some of the other places that nitrogen enters organic molecules or is available to the living sphere, the biosphere, are coming from some of the salts of nitrogen. So nitrate is NO3, nitrite is NO2. And both of these complexes are ones that are easier for organisms to modify biochemically. So nitrate reductase can take NO3 and electrons and make NO2, make nitrites. Nitrite reductase can take NO2 and more electrons and make ammonia. So nitrate reductase and nitrite reductase are very useful enzymes for increasing the availability of nitrogen to the living sphere. And they're obviously a lot less energetically demanding. these are also much more widely spread, so that plants and fungi and many bacteria are capable of catalyzing these reactions. They're also not poisoned by oxygen, so they can happen in ambient conditions. So when we're talking about nitrate reductase, you can take nitrate to nitrite. Nitrite can go through nitrite reductase to ammonia. This is the direction that is going to increase availability of nitrogen to organisms in general. There are competing processes. There are organisms that are using nitrogen as an electron acceptor, and those organisms go from ammonia to nitrate. This is nitrification. And in principle, some other organism could bring it right back. A third process that is sometimes taken advantage of by organisms that need electron acceptors and so forth is nitrate to nitrogen gas, denitrification. Denitrification is one that actually takes it out of the available sphere for organisms because we're back to molecular nitrogen. So this is sort of the sequence of denitrification. So if nitrogenase has created a fresh, new, expensive molecule of ammonia, how do we now get that ammonia into an organic molecule? And one of the very common places of entry for ammonia into the genuinely organic sphere is the enzyme glutamine synthetase. Glutamine synthetase is universal. Virtually all organisms have it. And it catalyzes a reaction that now genuinely makes things available for metabolism in other ways. So this is what that reaction looks like. You need a molecule of glutamate to start with, just like you need money to make money. So here's a molecule of glutamate. We invest in ATP to create a phospho residue here. And then glutamine synthetase can take that ammonia gas and displace the phosphate, so now you have glutamine. So you start off with a carboxylic acid of the glutamate, and by spending one phosphate bond, you've now put on the amine group. In addition to being a very powerful mechanism for making ammonia enter the biological sphere, it's also one of the mechanisms that's used for detoxifying things. That is, if you're producing excess of ammonia because you as an animal are eating more protein than you need at the moment, this is one of the mechanisms that's used for getting the ammonia out of just the ammonia state and into something that's organic so that we can get rid of it much further. When you look at the abundance of amino acids in most cells, what you'll find is that glutamine, the amide down here, that glutamine is much more abundant relative to the other amino acids than most of the other amino acids. And that's because glutamine is functioning as a nitrogen carrier. It's sort of the organically activated form of nitrogen that's widely available for other purposes. And so the enzyme that does this reaction is regulated in how its activity is going forward. On the other hand, if you look at that equation, you notice it does start with a glutamate. So where did we get the glutamate from? And this is also relevant to keeping the process going. If we've just created the glutamine to carry nitrogen, we're used up a glutamate. How do we get another glutamate? It's by a related or a metabolically related compound, glutamate synthase. And what glutamate synthase is going to do is going to use the nitrogen from a glutamine and a carbon backbone from the citric acid cycle. So here's alpha-keto glutarate, alpha-keto glutarate. It's carboxylic acids at both ends. And here's a molecule of glutamine. Glutamate synthase is going to use NADPH in order to reduce that carbon, which of course it needs to be an amine group, and move the nitrogen to that reduced carbon. So this is the outcome of the glutamate synthase, is we now have two molecules of glutamate, and now we can use that again with glutamine synthase, for instance, or for some other purpose. So the two enzymes working together, glutamine synthase and glutamate synthase, together end up fixing ammonia into organic molecules that are widely useful. Any questions? Okay. All right. Well, now that we've got the ammonia onto glutamate and glutamine, suppose we need something other than glutamate and glutamine. Well, it turns out that moving nitrogens around can be pretty inexpensive. There's a broadly named group of reactions called transamination. And transamination involves an amine group and a carbonyl group, an alpha keto acid. And the transaminase is able to just simply switch those two. So the transaminase can take the nitrogen from the amino acid and put it on the carbon of an alpha keto acid. So in this particular pair, what transaminase is starting from is glutamate, your 5-carbon amino acid. And here's pyruvate, an alpha keto acid. Does its transaminase thing. And now we have an alpha keto acid here, alpha keto glutarate again, and alanine, because we have an amino acid 3-carbon backbone. Glutamate is often the donor for the transaminase reaction. So it's one of the common carriers for this process. And often citric acid cycle intermediates are the recipients in order to make new amino acids. So transaminase is a way to make amino acids out of citric acid intermediates and other relationships as well. Transaminase uses another cofactor, one we haven't looked at before, pyridoxal phosphate. I don't expect you to memorize the details on this, but I think it's some good chemistry, so just sort of take a look at it. Here's pyridoxal 5-phosphate, and you can see the phosphate hanging off of there. Here's pyridoxine. That may sound familiar to you because it's vitamin B6. In the pyridoxine state, you have an alcohol. In the pyridoxal, you have an aldehyde up there that's been phosphorylated. And so this cofactor on the enzyme allows for this transamination reaction to be catalyzed for free. There's no energy input put in. The two state configurations are pretty equivalent. Transaminase is a good marker for whether or not tissues are intact. If you've damaged muscles or if you've damaged the liver, those organs can just sort of leak because they're damaged. And one of the enzymes that will leak out is a transaminase. So it's a simple reaction to test for as a marker for whether tissues are intact. So now we have nitrogen in a form that's biologically accessible. Let's think a little bit about amino acid synthesis. This is this overview roadmap again. Amino acids are needed to be the monomers that we would make proteins out of. We're going to be looking a little bit at how we can use intermediates to make amino acids. And after that, we'll look at the breakdown of amino acids to some extent. The carbon backbones that we are going to use to make amino acids come from several sources. Some of them come from glycolysis. Some of them come from the citric acid cycle. Some come from the pentose phosphate pathway. Those are the 3 commonest sources. The nitrogen that we're going to need for amino acids is frequently from either glutamate or glutamine. And we can still make 10 of the 20 amino acids. We call amino acids that we cannot ourselves make essential amino acids, meaning it's essential that they be in the diet or we won't have enough. The 10 that we can make are called non-essential, but that's shorthand for dietarily non-essential. They're every bit as essential for life, but they're so simple to make, we still make them. Or they're so important to make, we still make them, one or the other. And we're going to see that there's kind of a logic to which ones are which. Now, glutamine and glutamate, that's not too surprising. We just made some, didn't we? Alanine is very close to pyruvate. Aspartate is very close to oxaloacetate. Asparagine is close to aspartate. Serine is close to glycine. Glycine is very small. Tyrosine is not that simple looking a molecule, but it has that phenylalanine ring. And if we get the phenylalanine, we can make tyrosine from phenylalanine. So it's only non-essential if we're getting the phenylalanine in the diet. If we look generically at the kinds of molecules, the kinds of amino acids that are essential, that our bodies gave up making, you'll notice that they're the fancy ones. Tryptophan, huge big rings, 2 fused rings. Phenylalanine, phenyl ring. The branched chain ones, so valine is branched, isoleucine is branched, leucine is branched. Histidine, it's a heterocycle. There's nitrogens inside of a cycle. Arginine, that long, fancy thing down there, right? Lysine, again, a fairly long, complicated one. So there's a certain amount of logic as to which ones are essential and which ones we've kept the skill at making. So for the non-essential amino acids, how do we go about making those? Well, transamination, as I mentioned, can make several of them. So pyruvate from the glycolysis can make alanine. And if you think about the structures of these, it will make perfect sense. Oxaloacetate, 4-carbon alpha-keto acid makes aspartate. Alpha-ketoglutarate, 5-carbon alpha-keto acid makes glutamate. So those 3 can all be made by transamination. If we have aspartate that we've made from oxaloacetate, we can use asparagine synthetase. It'll take the nitrogen from glutamine and an ATP. It costs us a double high energy bond, splits off pyrophosphate to leave a glutamate over there. And now we have asparagine, you see, with down there. So asparagine synthetase uses the nitrogen from the glutamine's amide group. Glutamine synthetase can amidate glutamate to glutamine. And then, so what we're seeing is that from just a small number of citric acid cycle intermediates, we can get 5 amino acids by fairly simple reactions. Glutamate, 5-carbon backbone, provides carbon backbones for another couple of amino acids. It sort of makes sense that proline only has 5 carbons. And you notice by the dotted line, I'm not going to show you how we get to proline from glutamate. We've already mentioned that it goes to glutamine. But if you look at asparagine just has a 5-carbon backbone, really. It's got this funny guanidino group at the end, but it just has a 5-carbon backbone. So through enough steps that I'm using a dotted line, we can make arginine out of glutamate. I'll mention that the reactions for arginine are complicated enough that in young children, they actually don't make arginine quite fast enough. And it's very good for children to have arginine in their diet. But as long as you're getting whole protein, you're usually going to get some arginine. Serine is a little trickier. We're going to use 3-phosphoglycerate. So here's a second glycolysis intermediate that can give rise to an amino acid. So it's 3-phosphoglycerate. It's a carboxylic acid there. We are going to oxidize it. So this is an alcohol. We make a carbonyl carbon. We make an alpha-keto acid out of it. This is now an alpha-keto acid. 3-phosphohydroxypyruvate, actually. And glutamate is going to donate a nitrogen. And now we have 3-phosphoserine. And we throw away the phosphate to make serine. So glycolytic intermediate gets us to serine. Glycine does a couple of things. One of the ways that we can make glycine is from serine, using serine hydroxymethyltransferase. But more importantly, tetrahydrofolate. Tetrahydrofolate is another cofactor that plays a very important role in a lot of different reactions that involve moving one carbon around. So if you've seen folic acid, for instance, folic acid is the vitamin precursor to tetrahydrofolate. And it turns out to be very well adapted to moving a single carbon in various oxidation states. So this is one of the reactions by which we can get glycine. So if you have some serine, you can transfer this carbon to tetrahydrofolate, and that'll yield a glycine. But in fact, this reaction can go both ways. If you've got more glycine than serine, and you have some methyl tetrahydrofolate, you can make serine. So this is a two-way street. This is showing you tetrahydrofolate in a little bit more detail. This is the actual business end of it. This nitrogen, well, this nitrogen and carbon, like these two nitrogens, I'll say, these are the nitrogens that can carry the moving carbon. So we're going to move a carbon at different times of different oxidation states. That's the actual carbon that will be added or subtracted to things. But this double ring structure, this heterocyclic ring structure, is the tetrahydrofolate itself. This is the whole structure of it. You have here the pterin ring, paraminobenzoate, and then a whole bunch of glutamates. We're not entirely sure why tetrahydrofolate needs a glutamate tail, but there it is. Folic acid gets its name from Latin for folio or folium, so it's from leafy things. Leafy vegetables especially are rich in folic acid, but many vegetables have some. And it was in recent years that we've discovered that folic acid has an extremely important role in early development. Pregnant women in especial need to have enough folic acid in their diets because it will prevent a congenital birth defect called spina bifida that, for reasons that are not yet fully understood, in the absence of enough folic acid, an embryo is likely to develop with an incompletely closed neural cord. And so babies are then born with a partially open spine, which is a big nuisance and not much fun to live with. And folic acid prevents that almost entirely. It has cut the incidence down by over 90% by the simple matter of adding folic acid to many kinds of baked goods and so forth. But anybody who is in circumstances in which she might get pregnant, I won't say planning to get pregnant because people don't always plan to get pregnant, but in circumstances in which she might get pregnant might want to be sure that her diet includes enough folic acid. Another health consideration of folic acid is there seems to be a role in the health of the heart, that low levels of folic acid may contribute to elevated levels of cholesterol. And we don't know, again, quite all of that, but that's a growing public health story. So it may turn out that the supplementation that was undertaken to reduce spina bifida may, over time, also lower heart disease, but that's something that only time will show. Any questions? Okay. Cysteine. Okay. So if you look at serine, it's a 3-carbon amino acid, and you look at cysteine, it's a 3-carbon amino acid. It shouldn't be too surprising that serine and cysteine are kind of related for their synthesis, but they do it in kind of an odd way. So here we start with a serine molecule, and a homocysteine is an amino acid that is not used for proteins, and it's the same as cysteine with an extra carbon in it. When you hear homo this in biochemical terms, it often means almost the same structure plus a carbon. So homocysteine has a sulfhydryl group on it, and by a dehydration reaction, we can create a cystathione in which we have, in effect, 2 molecules of cysteine linked through this sulfur. I shouldn't quite say that. This is still the homocysteine. Anyway, we now have a sulfur bridge. It's not a disulfide. It's just a sulfur bridge. With a further reaction using water to split the bridge and removing the ammonia, we now have cysteine here, which has almost all of its structure coming from the serine with just the sulfhydryl group coming from the homocysteine. And then the carbon backbone that's released in that is alpha-ketobutyrate, not alpha-ketoglutarate, but alpha-ketobutyrate, which then has other reactions that take care of it. The essential amino acids, that is, the amino acids that are essential in the diet, typically have complicated and energetically expensive pathways for their synthesis. Free-living organisms have to make them all because they're out there in the wild. We usually consume things that have amino acids, so we've, over time, lost some of these pathways. And the way in which to keep track of some of these things is we cannot make branched chain amino acids. So that would be valine, leucine, isoleucine, or threonine. Those are all branched chain if you look at their structures. We can't make aromatic amino acids from scratch. So we can't make tryptophan, that huge, we can't make phenylalanine. We can use phenylalanine to make tyrosine, but not the phenylalanine itself. And we can't fix sulfur into methionine. We have to have methionine as the source of the sulfur for the cysteine. This is what these are reactions that bacteria can do. We can't do it, but bacteria can. They start from a serine molecule, so the 3-carbon backbone. They can invest an acetyl-CoA to activate that carbon in effect. So now you have this thing that looks kind of high energy-ish, right? And inorganic sulfide then can be attacked through another enzyme by throwing away the acetate group off here, and now you have a cysteine molecule. So rather different from the way that we do it. But this is a process to fix inorganic sulfur, sulfur that's just ionic. Land plants can use sulfate, and then obviously they have to reduce that. And reducing it will cost some energy. How do we make methionine? We can use homocysteine as the source of the sulfur. Methyl tetrahydrofolate is going to then add the methyl group to the end of the cysteine, the homocysteine, excuse me, to make methionine. This is one of the places in which it possibly folic acid is important to heart health, that when an individual has low levels of folate, this reaction in terms of transferring the sulfur over to make a methionine, or transferring the methyl to make a methionine out of the homocysteine, can lead to homocysteine accumulating in the blood. And there are individuals who have a genetic form of homocystinemia, meaning high levels of homocysteine in the blood, and those individuals are at much higher risk for heart disease. So, without going into all the details of how these various compounds are made, I think it's useful to sort of get a very broad sense of how some of this happens. So aspartate provides some of the carbon backbones for several of these essential amino acids, from methionine, threonine, lysine. Pyruvate can give rise to isoleucine, valine, and leucine in organisms that are capable of doing that. So we can see that these glycolytic intermediates and citric acid cycle intermediates are important for the essential amino acids as well as for the non-essential ones. The aromatic amino acids are fairly interesting as well, and because of their application in Iowa, I think it's worth mentioning some of them. Here's Roundup over here, and we're going to see why I'm talking about Roundup. So here is phosphoenolpyruvate, which we remember from glycolysis, kind of an energetic looking thing. And here is erythrose 4-phosphate. Erythrose 4-phosphate, of course, coming from the pentose phosphate pathway. That can give us a 7-carbon backbone here, right? 7 carbons. And if we invest another phosphoenolpyruvate, we can end up with a ring structure chorismate. So this is clearly edging towards being an aromatic ring, right? It's not quite there yet, but it's clearly an intermediate for it. So here we have one of the important intermediates into making the aromatic ring. Glyphosate, this molecule, is the generic name for the herbicide Roundup. And you can see how this has structural similarities to phosphoenolpyruvate. And yet, fortunately for us, Roundup does not inhibit glycolysis. So that's rather handy. And it might be worth reflecting a little bit on why perhaps glyphosate does not inhibit glycolysis. Tryptophan is a really stunning amino acid. So just to refresh your memories of what tryptophan looks like, you've got a fused 5-member heterocycle with your benzene ring here. So the biggest, bulkiest, weirdest side chain of them all. Tryptophan takes 13 steps to be synthesized from scratch. So it's not at all surprising that tryptophan is one of the ones that's most often lost in the evolution of metabolic pathways. The last two steps of tryptophan synthesis are actually occur on the same protein. So tryptophan synthase is a bifunctional protein that has two enzymatic activities in the single protein. What they're going to do is they're going to take indole 3-glycerol phosphate. So you can see sort of a glycerol group in there, indole 3-glycerol phosphate. It's going to split off a glyceraldehyde, so we're going to split off that group, leaving us an indole ring. And then the indole ring has a serine attached to it that creates it into an amino acid. Tryptophan synthase is rather cool because this indole ring, which is only useful really for this, sort of channels through the center of the protein to the second site where the serine will be attached. You can see here that there's a pyridoxal phosphate as a cofactor to it and a variety of other things as well. Histidine is a kind of a cool amino acid. Histidine is the only amino acid that gets nitrogen atoms from a nucleotide. So that's fairly different. Here we have a ribosyl pyrophosphate. We're going to see more of ribosyl pyrophosphate later, but it's a ribose pyrophosphate, phosphoribosyl pyrophosphate. Here is ATP, and contributing additional contributions come in from glutamate and glutamine. And what you can see on the board, I think probably not on your handout, is that these different carbons are all marked from where they've come from. So this nitrogen and carbon here, down here in the histidine, are this nitrogen and carbon up here in the ATP. This nitrogen and this nitrogen are coming from glutamine and glutamate. And this carbon backbone here, this 1, 2, 3, 4, these 5 carbons here are the 5 carbons of the 5-ribosyl pyrophosphate. So histidine is an expensive little amino acid to make. It's heavily involved in RNA intermediates, and some people have suggested that possibly histidine is a relic in some sense of an RNA world. Another characteristic that's rather relevant about all this is that histidine synthesis shares this compound, phosphoribosyl pyrophosphate, PRPP. It shares PRPP with nucleotide synthesis pathways. So this is one place where amino acid synthesis and nucleotide synthesis are joined together through a common intermediate. And we can suspect that that's maybe one way to try to keep the two of them in proportion to what the cell is likely to need. Okay. Now, interestingly, amino acids are intimately associated with the process of neurotransmission. That is, for nerve cells to talk to each other. You may remember when we talked about membranes and the synaptic transmission, how you dump neurotransmitters into the synaptic gap. Well, a surprising number of those neurotransmitters are amino acids. This maybe shouldn't be surprising. This may be a relic of when we were free-living organisms that were always looking for amino acids. But it's been adapted in a number of ways. There are some amino acids that are themselves neurotransmitters. They themselves actually act in the synaptic cleft to activate another neuron. So glycine, glutamate, and aspartate are all of them capable of triggering some, not all, neurons. And you have to have the right receptor, obviously, for it to act. So these amino acids are themselves neurotransmitters. But there are also a number of neurotransmitters that are derived from amino acids. So for example, if you take glutamate and you remove its carboxylic acid, you can make GABA, gamma-aminobutyrate. So here is a 4-carbon backbone. It had up there a carboxylic acid. That was when it was glutamate. And now we have gamma-aminobutyrate. And GABA is one of the major inhibitory nucleotides, ones that turn down certain reactions, so that it's important in the nervous system to have enough up and enough down so you're in the right spot. And GABA is one of the important ones for the downside. Tyrosine is the precursor to a number of important ones. So this is just refreshing of what tyrosine looks like with its phenyl ring and an alcohol. And if you decarboxylate it, you now have, and add an alcohol, you now have dopamine, right? So this is just the amine group up there, and 2 alcohols off of the phenyl ring, that's dopamine. And we call this ring structure here a catechol. And the derivatives of dopamine include norepinephrine and epinephrine. I'm sure both of those are familiar to you as adrenaline or noradrenaline. Dopamine itself is a neurotransmitter, as is norepinephrine. There is some thought that several of these compounds may play a role in depression. Certainly several antidepressants have the known biochemical effect of prolonging the life at the synapse of these compounds. And if you're interested in medical sides of these things, I can really heartily commend you a book called Awakenings. Oliver Sacks is a neurologist who writes quite beautifully about the human aspect of medicine. And in Awakenings, he discusses how the discovery of dopamine transformed the lives of patients with Parkinson's disease. Another amino acid that gives rise to precursors is tryptophan. So here's our complicated tryptophan again. And again, by decarboxylating it and adding an alcohol, we have serotonin. Serotonin may also be familiar to you as a neurotransmitter that may play a role in depression. Prozac is one of the reuptake inhibitors, so it increases the amount of serotonin at the neural synapse. Serotonin, after several more bits and pieces are done to it, can be made into serotonin into melatonin, and melatonin is a neurotransmitter that seems to play a role in circadian rhythms and the ability to track day and night and so forth. Okay. Yet another amino acid giving rise to a neurotransmitter is arginine, big fancy end of the molecule. So here's our arginine again, long fancy end. And it gives rise to an extremely simple neurotransmitter, nitric oxide. Nitric oxide. It's a radical, not an ion, but a radical. Nitric oxide is turning out to play a role in a very large variety of systems, muscles, heart, immune system. And we discovered nitric oxide by accident over a century ago. In 1869, it was discovered entirely by accident that nitroglycerin would relieve the pain of angina pectoris. So someone who's developing heart disease, who's at risk for a heart attack, one of the signs that that's coming up on you is that when you try to do too much, you get chest pains, angina, pain in the chest. And glycerin would relieve that. We had no idea why. And it turned out that glycerin breaks down to yield nitric oxide. So you can see here the nitrogen at this end of the arginine. We're going to oxidize that into hydroxyarginine, and then another reduction step. Sorry, we're going to reduce that. This is going to be reduced to this, reduce it again, and this time, both times, adding some oxygen in there. Very strange stuff. The citrulline here, we're going to see citrulline again when we talk about amino acid degradation, but this group can break off as a nitric oxide. So we've talked so far about synthesizing amino acids, and obviously they are very important to nitrogen-containing compounds, but the other big group of things that we have to synthesize that contain nitrogen are nucleotides. And I'm torn. Should we go ahead or should we hold it for Monday? I will point out that there are refreshments out there, people. Have a good weekend. AV